My name's Kai Miller. I'm a professor in neurosurgery here at Mayo Clinic. I am here at the world headquarters of STEIN, the Society for Technology, Engineering, and Innovation in Neurosurgery, and we are here interviewing Chris Honey.
And I'm Brian Clausen. I'm a movement disorders neurologist here at Mayo Clinic, and I work alongside Kai with our movement disorders cases. And it's great to have Dr. Honey here today.
I'm a computational cognitive neuroscientist from Johns Hopkins University, and I'm excited to be here today.
Great. Well, you and I have known each other for probably nearly 20 years. I think we first met at the Sagamore Hotel in upstate New York at a brain-computer interface conference a long time ago. And well, we've interacted ever since, sort of with a shared interest in large-scale brain dynamics, how the brain is organized, and how we can try to understand the way that different brain regions interact with one another using measurements, in my case, all from implanted electrodes in the brain, but in your case, with functional MRI. So it's nice to have you here visiting us. I guess, well, we can get started right away. But it might be nice maybe to hear a little bit more about you. You have one of the more interesting life stories, amongst different cognitive neuroscientists.
Well, I guess I grew up originally in Namibia in Southern Africa. I did my high school in Johannesburg. I got my undergraduate degree at the University of Cape Town. I suddenly became aware of cognitive sciences and the neurosciences and the fact that one could get paid to do a PhD in the United States. And I started a graduate program at Indiana University where I began to discover neuroscience. I studied with Dr. Olaf Sporns, who is an expert in brain networks, and this is where large-scale brain dynamics first entered my awareness. And then for my postdoc, I went from doing more abstract modeling type studies to studying some of the most complex brain responses that we can measure, which were responses to things like stories and movies, while at the same time trying to characterize large-scale brain dynamics in those settings.
And well, moving back a little bit, you didn't start out in neuroscience. I mean, you're one of those rare individuals where, if I need somebody to tell me if I'm right or wrong on the math, you're the person.
My undergraduate degrees were in math and applied math and in literature. And that was something that actually drew me to the neurosciences, which is that I'd always felt this kind of draw towards science, but also towards the humanities, and then the neurosciences are something that is a kind of a merger of the two.
And well, I mean, your parents mathematicians as well?
No, my parents were journalists when I was growing up. They were journalists in Southern Africa, so they covered a lot of the transitions that were happening, the end of apartheid in South Africa, the end of the rise of the onset of independence in Namibia. So it was a pretty eventful and I feel very lucky to have had that childhood in a way, to have experienced those changes.
Yeah. Well, I think something that interests the two of us is this idea of intervening with the brain. That's what we do with our patients, by putting electrodes into brain areas. And a lot of the focus, particularly in the media, is around things like brain-computer interfaces, where there's one particular area. So areas, in a very concentrated part of the brain surface where people put a ton of electrodes and examine, let's say, a specialist area for one thing or another. But I think one of the lessons that you've taught me over the years is that brain networks are truly distributed. And we have areas that can look a certain distance into the past, and you've actually shown that as we stitch together our perception of reality, there's no one place that does do this, nor one place that has the computational capacity to. But we stitch together our illusion of reality based upon, why don't you tell us, based on?
Well, let's take a step back and think about some terms that we are familiar with, something like short-term memory or working memory, right? So if you ask the question about how do we hold in mind information from the recent past, right? Think about you are traveling and you're at a new Airbnb, for example, and you have to you ask somebody what's the Wi-Fi password, right? And they tell you, the Wi-Fi password is like 6QZ53, right? And you have to hold that in your mind. And there's some sense in which that information that you just heard was from the past, but you have to kind of keep it available, right?
6QZ53.
Great.
If I don't keep saying it, I'm not going to remember it.
But sometimes when you go to an Airbnb, the password is 65QZ3 exclamation mark underscore 3394LPQ 6. And in those cases, we suddenly become aware that there's a limitation in what we can hold in our minds, right? And so a lot of the work in the 20th century in kind of psychology and in cognitive neurosciences was kind of informed by this idea that we had something like a store for holding information that was a limited capacity store, and you can feel the limitations in capacity sometimes, right? When you can feel you can't fit anymore, right? And so there was this trend to kind of treat that store as the thing that we use for stitching together our experiences in life over time as well.
Well, the idea of building out some spatial structure that we have a physical interpretation and store our memories there. I mean, this goes back to the ancient Greeks, right? They had this idea of a memory palace, and they would fill the memory palace with objects. And these are people who could remember, amounts of information that would dwarf anyone alive today.
Right. So we know that in those cases, the memory palace, for example, that, there are some edge cases here, but we know that things like using the method of loci, where you will store the digits of pi in different locations in a room, that's dependent on having a hippocampus, right? So if you don't have a hippocampus, you can't do that. Whereas the case that I described earlier with the Wi-Fi password, people without hippocampus can do that. And so that distinction is part of one of the most informative and kind of influential discoveries as well of the 20th century in the study of memory, right? And you are familiar with the cases of HM and other famous amnesic patients, right?
Sure, yeah.
So
HM, do you remember HM?
Yeah.
So bilateral hippocampal removal, lost ability to form sort of explicit short-term memories.
who was the surgeon?
I don't know.
that.
It's Canadian. Yeah.
Scoville?
Yeah. Henry Molaison. I don't remember the name of the surgeon. I should.
So HM, right, Henry Molaison, he would be totally fine if he was around today, which unfortunately he isn't, but if he were around, he would be able to hold the Wi-Fi password in his head, right? Mm-hmm. There was this sense, getting back to what you said earlier about kind of information being kind of stored in one area or one kind of function, there was this idea that there's some particular part of the brain, perhaps some subset of the prefrontal cortex, that is the location where you store information from the recent past that you want to keep active, right?
Mm-hmm. Sure.
And so there are all kinds of tasks that people have done and work in both humans and animals, right? That have validated this idea to some extent, right? That it is the case if you have lesions in the prefrontal cortex, then you can't hold that information in your mind anymore. But there's this question, which is that the only way that we hold information in mind or we keep information available over the course of, let's say, 15 or 30 seconds in our lives, right?
Sure.
And is there one thing that we call short-term memory or working memory that's localized there? And one of the things that we started to think about in our research when we started studying movies and stories is when we started looking at it more closely, it just seemed like there was just way too much information that we needed to be keeping around or integrating over time in order to understand the story, or this conversation that we're having now.
Sure.
It just seemed like the content of this conversation is just way beyond the Q6 VC3R, Wi-Fi code. And so one idea that my postdoc advisor, Uri Hassan, and some of his colleagues had started to put forward was this idea that there was a distributed cortical representation a distributed cortical processing in which memory was inherent, essentially, and that this distributed processing had a kind of a characteristic time scale to it, with short time scales in the sensory cortex and then increasingly longer time scales.
When you say sensory cortex here, you mean occipital, early V1, V2, that kind of thing?
Well, for the Wi-Fi code, it would be auditory cortex, but
sure.
if for watching a movie or something, it would be
Yeah.
So in fact, a relatively early auditory area and a relatively early visual area, generally we wouldn't expect them to have that much memory capability, but they would each have their own ability to integrate over time. That was the idea. They're not going to be taxing that prefrontal circuit when they are making use of information from a second or two earlier.
Now, did you guys do, I mean, in my mind, I remember you playing, hopefully you can get to it. I think you've prepared some slides. Did you guys look at the auditory system as well as the visual system? In my mind, it was always visual, but I guess that reflects my own
Pretty wide
scientific bias.
coverage from the, yeah.
Yeah, so we've done both visual and auditory, and this phenomenon is found in both pathways. So, what we see on this slide over here is just a lateral view of the human cerebral cortex, and there's some familiar landmarks.
Sure.
The earlier auditory cortical areas, although they're actually tucked into the Sylvian fissure,
Sure.
but we can see them with a blue
transverse
circle.
temporal gyrus.
And then as we step, so one thing that we know, right, and one thing that we get taught when we learn neuroscience is this notion of hierarchy, right, in the brain. And the visual system is where this kind of came from originally. But there's a similar kind of story in the auditory pathway. And the story is something like, in the sensory periphery, there are these low-level kinds of features that circuits and neurons are sensitive to. And as you move deeper and deeper into the brain, or further along this auditory processing pathway, the circuits become responsive to, they extract more abstract features and more conjunctive features. But one of the things that we were claiming was that they also extract patterns that extend further over time. Okay. So that by the time you step a couple centimeters posterior of the middle superior temporal gyrus, you start to find areas like in that green circle that are going to be sent. Their response at one moment in time is going to depend on the content of the stimulus from a few seconds earlier, whereas sites in A1, for example, are going to show a much smaller sensitivity to the content of the stimulus from 3 seconds earlier.
Sure. A1 being
primary
gyrus,
primary
transverse
auditory
temporal
cortex,
gyrus. Yeah.
Yeah.
Sure.
So there's this funny kind of correspondence between this traveling from the exterior world where the sensory information arrives deeper into us. There's this correspondence between how far deep into our brains the information has moved and how much history sensitivity or memory the circuits start that they have. So coming back to this example with the Wi-Fi password, for example, right? Even this conversation, it's almost as if there are two ways, well, there are multiple ways, there's more than two ways, but there's at least two ways that we're storing information for the short term. One way is that if we are really attending to that Wi-Fi password, there are going to be some kind of patterns of activity in prefrontal cortex that we know are important for being able to repeat the Wi-Fi password back to somebody.
Okay.
But
is that because of the recruitment of speech or because of the way that you understand what you remembered in the context of what's going on with that individual you're communicating with?
So these prefrontal circuits that have this ability to kind of maintain information, they are thought to be fairly generic. So there's been some back and forth about this in the literature. So maybe the different subsets of prefrontal cortex might be preferentially involved in maintaining different kinds of information. So Leslie Ungerleider and some of her collaborators worked on questions like this. But it's still broadly a kind of memory that is, you can think of it as control-based. In other words, the prefrontal cortex is doing something to kind of keep information from the recent past alive actively, in some sense.
Sure.
And maybe different parts of the prefrontal cortex, sometimes maybe even premotor cortex, could be involved in doing this. But the kind of function is like a protection from interference. So there's this idea that information arrives from the world, it moves deeper into our brains, and then information that arrives later. So when you hear that Wi-Fi password, afterwards somebody starts talking to you, that new information is going to, in some sense, interfere with or overwrite what you just heard. Yeah. And so the original idea was that the prefrontal cortex would be protecting that information from interference. It doesn't get overwritten by the new input. But there's actually a parallel kind of complementary system in our brains, because in some cases you want the past information and the new information to interact with each other. So you don't want it to be protected from interference. You don't want it to be kind of held separate. And one example is like this conversation.
Sure.
You want it to be the case that the things I say at the end of a sentence are somehow mingling with the things that I say at the beginning of the sentence, right? And that kind of function is what we are claiming, and what the data seem to indicate is being distributed across these cortical hierarchies. Now, what we don't know still is we don't know exactly how. So we're able to map.
So how are you able to map? How did you discover this idea that you have essentially information from different timescales in the past being held? Well, I sort of know the answer.
And what inspired you to look at that initially? Were you sort of empirically seeing these trends in data that you were collecting for other reasons, or did you go out to actually look into this question?
So when I started studying it, there was already an indication of this being true in the visual system. So Uri Hassan and others had done studies where they took a Charlie Chaplin movie and they measured the responses to the movie when the movie was intact or when it was broken into small pieces that were, small segments that were reordered. And what they found was that the responses in visual cortex were largely unaffected by whether the story was scrambled or not. But as they moved deeper along the visual hierarchy, they found that the responses became more sensitive to scrambling of the content. And the idea was that the interpretation was that each patch of the brain has a kind of temporal receptive window. And that window is short in early visual cortex. And so changing things outside of that window, let's say the window is 1 second, changing things 5 seconds ago isn't going to affect the response of the circuit. But then in an area like somewhere in IT cortex, the window might be longer, maybe
IT meaning inferotemporal?
Inferotemporal cortex,
Sure.
I'm sorry, yeah. So there was already an indication of this being true in the visual system when I started studying it. And when I started studying it, we were using intracranial recordings, actually. There's a whole other side to that I think I want to come back to, but let me just give you one example of one way that we've also recently been showing this in the auditory pathway. Can I show you? So
Sure. Yeah.
Yeah, great.
this is a study that was kind of led by a former grad student in my lab, Sherry Chan, and it's a really fun kind of study. So what we did is we imagined two groups of people. These two groups at one moment in time are going to start hearing exactly the same speech, but before they hear that speech, each group of people is going to hear different speech. So there's a certain moment in time where they start to hear the same thing, and before that moment in time, they're hearing different things. Mm-hmm. And our reasoning was that if a piece of cortical tissue has, let's say, a short memory, mm-hmm. then that patch of cortical tissue is going to start looking very similar across the two groups right after they start hearing the same stimulus. But if a piece of cortical tissue has a longer history dependence and a longer memory, it's not immediately going to look the same across the two groups after they start hearing the same input. It's going to take a longer time for the responses to actually converge across the two groups. Does that make sense?
Yeah.
It does, but I would also say that doesn't the way that you prime somebody, or doesn't the way that you would give them information ahead of time bias the way that they're going to interpret this new information that's coming in?
So at a cognitive level, that is definitely happening. And what we're saying is that at the neural level, so we can measure this in different parts of the brain. So in some parts of the brain, even though people may have different expectations about what's going to happen next, it just, even so, that it doesn't affect the neural response.
Sure.
Yeah. So we can come back to this top-down issue as well. That's a good point.
Yeah, sure.
So does the design basically make sense? Yeah. So for each patch of tissue, we're going to measure how similar are the responses across these two groups of people. And we're going to map that as a function of time right after the onset of the shared stimulus.
Okay.
And what we found was that if we map that as a function of time, so on the y-axis here,
And these stimuli are auditory or visual?
these
or
are
both?
auditory stories.
Okay.
And what you're looking at in the area is broadband power
So
change?
this
or-
is actually a functional MRI study.
Oh, this is
Yeah.
MRI. Okay.
But so we no, there actually has been an intracranial study that
Okay.
Sam Norman Hagner actually and Nima Mesgarani have actually done a kind of a similar kind of paradigm using intracranial recordings. I think a couple of years ago.
Okay.
So when we introduced this, what we found was that in areas like in the middle STG, the alignment across the group starts out low and then it ramps up.
Temporal gyrus.
The middle superior temporal gyrus. Yeah. The alignment across the two groups. So the alignment across the two groups always starts out at 0. And because of hemodynamic effects, the ramping up, the alignment is quite slow. But you'll still see that it's the alignment, the ramping up, is the fastest in auditory cortex, the more auditory cortex. But as you step backwards, look in the right hemisphere in particular, you can see the steady progression from dark red, indicating a rapid ramping, to orange to yellow. And the yellow is the slowest ramping. So what you're seeing there is these two groups of people are hearing exactly the same stimulus, but the response is completely different for 7 seconds after the onset of the new.
I see.
So they're hearing exactly the same material for 7 seconds and producing completely different responses, but
So
the
this
responses
is
within each group.
alignment in the measurements you're making from each person's brain.
Correct.
And here IPL is inferior parietal lobule.
That's right. Yeah.
So that alignment, saying that aligns, assumes a couple of things. The first is it assumes that you can take the organization, the surface organization of two people's brains and map them onto one another.
Yeah.
And that the way that they process information, given that structural map on the surface of their brain, is going to be the same.
Yes.
But we know that for many people, there's, I do a lot of awake brain mapping. Dr. Clausen and I do a lot of awake brain mapping even in the thalamus, and there's variability across people for the precise place that they process a specific type of information.
Although you measured that, didn't you? Because you had multiple people in each of these groups
Yes.
exposed to the same.
Yeah. So so within each group, we can people have identical responses, identical stimuli, and we can measure the variability in the responses within each group. And so then we can always reference this effect against that. But the other thing that we can do is we can run this experiment within participants. So we can actually repeat the same material with one context and with another context. And you get basically the same kinds of results when you do it that way. When the material is very, very meaningful, you run into problems with that method because people can actually really remember. Then they use their long-term memory to remember the last time they heard the material. So to do that, you need to expose them to a lot of material, so they can't remember all of it very easily.
Okay.
But within subjects, with a lot of these experiments, we've done within subjects and between subjects versions of the experiment.
Okay.
But you're absolutely right that the kind of neural response that we're looking for alignment in here is relatively coarse. It's on the scale of some kind of a pattern, on the scale of a few millimeters. Patterns of activity spatially with so a region on the order of a couple centimeters across, and with maybe the fluctuations are the spatial fluctuations are on the order of a few millimeters inside of that pattern.
Okay.
Anyway, so this is just one study. There are actually many kind of variants and other kinds of approaches to doing this. So I'll just say, so you can do this with auditory stimuli. You can also do it with movies, silent movies. You can also do it with music. Now, one of the interesting things about it is that this study has been done, you can take like Brahms, for example, and you can scramble Brahms and break it into different pieces, different scales, and you can ask which parts of your brain are sensitive to the fact that this part of Brahms is actually preceded by the correct, what Brahms actually composed versus some other chunk of Brahms music that's been swapped into that place. And you see a kind of a similar kind of hierarchical organization where there's a gradient from early to higher order regions, but it's more compressed. It's not exactly the same areas typically. And another thing that's probably relevant here is that this study involved students at Juilliard, which is an important point. So when we talked about this other kind of this kind of memory that we use to integrate information over time that's different from the Wi-Fi password where you're effortfully holding it, that process requires you having some kind of knowledge, as you were saying earlier about expectations, right? So if a person like me who's not very knowledgeable about music probably wouldn't show the same effect that these Juilliard students are showing. And so this is one big difference between kind of conventional working memory and this kind of distributed memory that we've been mapping, is that this distributed memory actually is very knowledge dependent. Similarly, if I presented a story to people in a language that they don't understand, they're definitely not going to show this architecture, right? So this architecture in some sense reflects how it's a knowledge dependent or learning dependent kind of memory.
So the life experiences that they've had will have given them either a personal scaffolding to place these new experiences into, or, I mean, is that scaffolding shared between experts?
Well, it looks like in the case of these Juilliard students, it is. And some people think that, there's some theories of interpersonal communication that are that imply that we've selected some aspects of our language organization because they're the kinds of things that tend to align across people, if that makes sense. In other words, language in some ways is set up to take advantage of the fact that we have similar kind of biases, right, in the way that our neural systems infer things or learn. Does that make sense?
Yeah, it does. Although I wonder, let's say you have somebody who grew up in East Asia and learned to write with kanji initially, and then their language architecture presumably would have fairly significant differences.
Well, so.
And then they went on to become an expert in something that we're saying is a proxy off of that language architecture that they learned in the form of music, unless music's really its own thing.
So I actually don't know a lot about the music to language correspondence, but my rough impression is that the people who've studied this in the last decade indicate that the music and the language pathways are actually somewhat distinct. So when you look at the group level, they're very, very nearby each other, but when you look within single subjects and you more carefully map it, the kind of areas that are really have a peak response to musical versus linguistic are often nearby, but not overlapping.
Topologically distinct.
Yeah.
Sure.
Well, I want to get us to the intracranial recordings a little bit, if that's okay.
Absolutely.
So part of the reason that we were doing the intracranial recordings is because we wanted to understand the temporal dynamics.
So, of course, I know what intracranial recordings
sorry.
are, and so does he, because that's what we do. What do you mean by intracranial recordings?
So, normally when we record electrical signals from people's brains, we place electrodes on the outside on their scalps, right? And those measurements are easier to make, but you are not able to get a really high quality measurement of the electrical signals in the brain from that.
EEG.
For example, EEG. Sure. But there are certain situations where, and they're generally clinical settings, where it's possible and the patients are willing to allow us to run experiments while they have electrodes implanted for other purposes.
Sure. Yeah. And so that's what we do. We place these very often either for monitoring to figure out where patients' seizures start from. In some experimental contexts, it's trying to understand psychiatric networks patients will have. Or we place electrodes to basically stimulate the brain long term and help rescue function in the case of misbehaving brain circuitry. Yeah. So intracranial recordings, I guess, in this context, you mean either wires that are placed into deep structures throughout the brain or electrodes that are placed on the surface of the brain. Mm-hmm. Mm-hmm. Okay. Sorry. Yeah.
So and in the case of the experiment that I was doing when I first started studying this with intracranial measurements, it was grids of electrodes implanted over here on the surface of the.
And that, of course, is how we got to know each other.
Yeah, because
we were both doing this together.
Yeah. So Kai Miller essentially taught me how to analyze that kind of data. It's also worth mentioning in passing just that the patients are extremely generous with their time, and they just I was when I first started doing this work, I was really just surprised at what a large proportion of these patients in the hospital, they're going through often a difficult time, and they're willing to take time to help us and to participate in the research.
Sure. Yeah, absolutely.
So there's the study that I was mentioning before, where we were, showing two groups of people the same story, right? And you were mentioning that, so those were functional MRI responses, which are measurements of blood oxygenation change, and they tend to be kind of a sluggish, blurry index of brain activity. But with intracranial recordings, you can measure, 1,000 or more samples per second of electrical activity from populations of neurons. And because of that, it provides a much more precise measurement of how brain dynamics are changing over time. So one of the things that we found with the intracranial recordings when we did a similar kind of experiment was we found that the speed of fluctuations in neural population firing. Now, so this is something that I learned about also more than a decade ago, is that with these intracranial recordings, another kind of distinctive advantage that they have is that they provide you with a way of estimating the firing rate of populations of neurons underneath these electrodes.
Sure. Yeah.
And so we estimated that while people were listening to stories or while they were just at rest staring at a dot on a screen. And what we found was that the fluctuations in firing rate were faster in some areas and slower in other areas. And so then we had this
the fluctuations. So does that mean the time scale? So here you have one that's every 50 milliseconds is widely different, and there you have one that every let's say second is different. So if you look at how similar a signal is to itself at later points in time, is that what you mean, or what is it that you mean?
Let me give you an example. Yeah. So if you look at on the bottom left over here, you'll see fast fluctuations of power, and on the bottom right, you'll see slower fluctuations of power. And those fluctuations of power are our estimate of the changes in the firing rate of these neurons, or the rate of spiking of these populations of neurons.
Yeah.
And so some areas look more like the bottom right, and some areas look more like the bottom left. And we asked this simple-minded question, which was, is there a relationship between this just speed of going up and down and the memory, the kind of the memory in these circuits?
And by memory, you mean quantified by how much,
Yeah.
how long that
How
movie
history dependent they
or
are, right?
story would go before you would scramble it.
Exactly. Basically, so the idea was, if a brain region is more sensitive to things, if its response in right now depends on things that happened 5 or 10 seconds earlier in the story, is that kind of a brain region going to have slower dynamics compared to a brain region in, let's say, in primary sensory cortex, which tends to have a very small dependence on what happens 5 or 10 seconds earlier.
Okay.
And in that study, that's what we found. Okay. And we found that there was actually a very, very close relationship between these two things. So as you move now from the sensory cortex along the cortical hierarchy, you find these two things happening at the same time. The population firing
Along
rates.
the cortical hierarchy, you mean further down this, or remembering more and more periods of sensory data? So remembering bigger and bigger chunks of sensory data?
Yeah, so something like, coming back to our hierarchy over here, the areas in blue are the more kind of closer to the primary sensory cortex.
Shorter memory of the outside world. Okay.
And those areas, we've typically thought of them as being responsive to things like, vocal sounds, for example. And those areas don't have a large amount of memory of things that happened many seconds earlier, right? So these are not absolute statements, because it's kind of a graded change, but their dynamics are faster than the dynamics that you see. In other words, the fluctuations are faster than those that you see in the green and in the red circle. And so the red circle is this part of the brain that isn't even sensitive only to auditory input. It's kind of this, it's called a higher order association cortex that combines information from multiple modalities, vision and auditory, for example. And what you find there is that the responses depend on what happened many seconds earlier, and the dynamics there, the neural population activity is just slower, just on average. So there are a couple of important caveats here. Every brain region is capable of rapid changes. So we're not saying that, activity is always slow in these areas. It's just kind of a tendency on average that the dynamics are slower. And states the neural population can remain in a certain state for a longer period of time. Yeah. So that's one of the things that we discovered using intracranial recordings, is that there's this correspondence between this functional property of our brains, which is about how they handle information, and then this physical property or kind of biophysical or physiological property of circuits, which is just how rapidly the firing rates change.
Okay. So I guess something that I'm interested in is if we're designing devices that are going to try to, let's say, decode perception. We had some work where we decoded very early perceptual things, faces and houses and tools and that kind of thing. But let's say we really wanted to be able to decode the human perceptual experience. Do you think your work is telling us that our devices have to be recording from many different areas of the brain at once, not just getting really high resolution from one particular brain area? Or, in other words, how much is our stitching together of the context we're living our lives in a distributed process versus, you have a series of movies here, a series of movies here, a series of movies here, and those are always being compared against one another? Does that make sense?
I think it makes sense. So, I guess we should just start with the empirical, what we know empirically, which is that when you try to build decoders with fMRI, for example, if you try and build decoders with fMRI, the best decoding comes from pooling information across multiple parts of the cortical hierarchy.
Okay.
So when we, so I guess it's worth saying something, which is until a few years ago, maybe 5 or 10 years ago, we didn't have a way to predict brain responses across all these stages of the cortical hierarchy to something as complex as a story or a movie. We could predict things in the relatively early auditory pathway and maybe in a couple of areas that we knew were sensitive to a particular kind of feature. But if something was happening like, I don't know, 2 characters in a story were starting to talk about baseball,
Sure.
we just didn't know how to map that onto what is going to happen in the brain. And then as of, around 5 or 10 years ago, actually we started to be able to do that using tools from machine learning. And so we would insert these things like stories and movies containing things about baseball into one of these machine learning models, and we would use the interior states of those models as features which we would then use to predict the response in the brain. And to a very surprisingly large extent, it worked. So we didn't necessarily understand what these models were doing or how they were representing the concept of baseball, but we were suddenly able to do this. And then those same models you can invert and you can use to decode when somebody is thinking about baseball. Right?
Sure.
And just in general, the ability to do that kind of decoding is superior.
So when you say you can decode somebody thinking about baseball, you mean if you, like thinking about baseball versus thinking about a music video they saw, or thinking about baseball and thinking about soccer?
Well, so I think we're still in the process of figuring out exactly how precise the decoding is. For any given category, like for example, baseball versus soccer, we could absolutely design an experiment where we could build a classifier or build a tool that could distinguish whether someone's thinking of baseball versus soccer. Absolutely. We could do it with a high accuracy. Although maybe I don't know what the accuracy would be on, given just like one sample of data in one person, like real-time decoding, but it would definitely be successful. But the kind of decoder that people are trying to build is one that is just open-ended. In other words, it's not like it's just trained to decode baseball versus soccer. It's trained to just take brain activity and just from the brain activity just tell you, like in words or in pictures, what this person is thinking of, or what their inner speech is, for example. So we know we can do it above chance.
I'm glad no one's done that with my brain.
Well, so there is, and this is not something that my lab
My
is,
wife might want
is
something
leading
like
in.
that.
Yeah, this is, there are other labs that are really pushing this forward. This is
Sure.
But the reason I mention it is just that empirically, that kind of decoding does pull signal across multiple parts of the brain. It pulls signal across these distributed circuits. There's a related point here, which is the question of how should we think of what is happening in this cortical hierarchy? What is the nature of the representation in some sense? And there's this idea that's not a new idea, but it's kind of recently become more influential, perhaps because of the connections to machine learning and our ability to build models like this. But there's this idea that our cerebral cortex, the hierarchy, is what's called like a hierarchical generative model, right? Okay. And so the idea is that if a part of the brain receives a certain kind of input, it learns also how to drive the layer below to regenerate a similar kind of input. So if you imagine a series of stages, like in this image over here, right? You got to go from the blue to the green to the red. Mm-hmm. The idea is that a forward pass signal from the word areas to the scenario areas. Scenario areas learn how to send a signal back to the word sequence area to regenerate a particular pattern of words.
So it's not just that we have brain structures that are trying to predict what the physical state of the outside world is. We have brain structures that are trying to predict what the state of earlier brain areas is. So there's a continuous sort of prediction machine.
Yeah, something like that. Yeah. So
I mean, is this a neural correlate of backpropagation?
When we build these models, these kinds of models in computational neuroscience, we do use backpropagation, but to the best of our understanding, especially for the case of backpropagation through time, which is what's needed to learn temporal models, we don't think that the brain is doing backpropagation. There are ways to approximate backpropagation when it's not backpropagation through time, but backpropagation through time, approximating that so backpropagation is this tool that's used to update neural networks so that they better perform the input-output function that they're supposed to perform.
Yeah.
One of the reasons it's considered plausible as an explanation of how the brain is learning to do that is that it requires the one part of the brain to know about essentially errors in other parts of the brain and weights in other parts of the brain. Mm-hmm. And so for at least maybe for 30 years, there have been debates about how plausible is it something like this is happening in the brain. But we do think broadly that something like a generative model is being learned. We are not exactly sure how. There are ways of building these toy versions of these models that work, but getting it to work with the kind of the richness and the spatial and temporal complexity that we know that our brains are doing, we don't know how that model that we have is being learned.
Well, very interesting. I'm very grateful to have you here. And anything you'd like to ask us about, or anything we should be asking you about in terms of how your work would directly relate to creating implanted constructs for helping patients?
Yeah, so there is one area that I think is a potential area of intersection between these lines of work, which is when you have these parts of the brain that are integrating sequences over time and they're kind of making use of this memory, they also have to have a way to decide not to integrate. They have to have a way to kind of forget. So for example, if somebody finishes one story and starts a new story, you kind of want to clear things out, right? Clear out your memory. And one possibility is that with electrical stimulation, we might be able to understand what are the electrophysiological processes that actually are performing that resetting or kind of clearing out of the internal state that these circuits have. So we've discovered that there's this distributed memory, but we don't know how the distributed memory is controlled. And we think that it's necessary to have processes that stop this integration process.
What would be your best guess at a site to explore with stimulation?
Well, so one possibility is that it would be like a neuromodulatorily, so for example, maybe releases of acetylcholine, for example, might be, right? So maybe stimulating the basal forebrain potentially. Another possibility is that some kind of processes that are used to separate action sequences in the striatum there may be kind of correlates in the more sensory pathways. So I would.
Something that interests me is the idea that we'll be able to create constructs to help people after they've had strokes, to help them have some kind of reorganization. And you can imagine in this tableau of areas that you have where they're integrating information over different periods of time, and there's a hierarchy where you have just the very early sensory all the way up to, remembering the long arc of a story of one kind or another. You've knocked out a piece in the middle there. What happens? What happens to this stitching together of these different time scales when part of it is lost?
So one possibility is that the timescale effect is an emergent effect of just this learning and generative model. So in other words, let's imagine that a higher order part of the brain is learning about faces and how they move over time and how people's facial expressions change.
Sure.
And there's a lower part of the brain that's detecting things like, where is this person's edge of their eye, or how is their head tilted, or whatever. And so that higher order part of the brain might end up having this greater ability to integrate over time simply because that's what it needs to do its job of being a good generative model. And it could be that just in general, more extended things and more abstract things require this. So an edge of your eye tends to be something that is just less stable in the world relative to like whether you're there or not. So abstract things like Brian and Kai are here. An abstract thing like that, a part of the brain that's involved in that kind of representation, it just it necessarily needs to be kind of tracking information over longer timescales. And so what I'm trying to say is that the underlying thing could be just that the higher order region is trying to learn from the lower region. So in the case of some kind of a stroke, for example, if you could get the higher order region so imagine you have 3 levels and level 2 is damaged. If you can get level 3 to now learn from level 1 instead, somehow bypass the signal from 1 onto 3, and then somehow facilitate learning in 3, in theory, level 3 should be able to actually compensate in some way, right?
Sure.
So I think that like the idea of a kind of relatively flexible general learning algorithm that allows any patch of cortex to learn these statistics and the ability to kind of regenerate its own input, if that's happening, then when the input gets damaged, in theory, if you could reroute past that area,
Sure,
you might be able to recover the function. But it would require knowing about the hierarchy, knowing what is the next stage in the processing pathway.
well, that's why we're talking to you.
Yeah.
Yeah.
Oh yeah, that's true. And so in theory you could use this kind of mapping to figure out which stages need to be linked. Yeah.
Sure.
Yeah.
Okay. Well, thank you so much for coming. We hope to have you again at some point in the future.
Oh, thank you.
Thanks a lot.
Thank you.
I know you and Kai have known each other for a long time, but I'm just meeting you. It's really good to meet you.
Oh,
Thanks for coming.
thanks a
All
lot.
right. Thanks a lot.